In this video, we're going to show how to run a pathway analysis using string database on Cytoscape. Gene set enrichment analysis is a critical component of high-throughput biological studies, providing key insights into the biological characteristics of a dataset. These analyses identify essential aspects such as biological processes, pathways, and phenotypes that are important for understanding the biological implications of the experiment. In the previous video, we showed how to run a DSIC2 analysis using GO2R, where we compare 20 samples of women with Alzheimer's disease to 20 samples of control or disease-free and obtain 2,530 differential expressed genes using a cutoff of full discovery rate p less than 0.05 and log2 fold change less than 2. So we will use this list of differential expressed genes to run an overrepresentation analysis or pathway analysis. So how overrepresentation analysis work? This analysis uses the Fisher exact test to find if our differential expressed genes are enriched in certain pathways or functions. In other words, we will compare our differential expressed gene list with curated pathways gene sets, for example, Kyoto Encyclopedia of Gene and Genomes, Reactome, Gene Ontology, Biological Functions, etc. One term that you're going to use frequently when you use a tool for overrepresentation analysis or pathway analysis is the reference set or background set. So this is going to be all the molecules that you detected in your methodology. For example, if you use a microarray platform, right, you're going to use a microarray platform IDs as a background set. Or if you use a proteomics, you're going to use the results of your proteomics, all the proteins measured in your analysis. Or you're going to use a whole genome if you use RNA-seq, for example. Essentially, it's all the molecules that the user detected in the experiment. So the user list should be a subset of this reference set, and it's going to be set by arbitrary cutoff of significant and fold change. And we're going to use pathway databases or gene ontology databases to see if those are enriched in our user list by using a Fisher exact test or hypergeometric test. There are different methods for pathway analysis. For example, one can run differential analysis, set a cutoff of full discovery rate FDR less than 0.05 and log2 fold change 1 and run an overrepresentation analysis with one of the many of available tools free online. Another method is to use all the differential analysis results with no cutoff and run a gene set enrichment analysis. These are different flavors of GSEA, and we show those in other videos. Other method is to analyze the interaction network between the genes, for example, using extreme protein-protein interaction network or analyzing the expression correlation network as in the weight gene correlation network analysis, which we also show in a future video. In this video, we will use string database to run an overrepresentation analysis and will cluster the resulting enrichment pathway to find overarching biological themes. So what do we need? Just two things. One, the differential expressed genes, which have been previously applied a cutoff of a full discovery rate p less than 0.05 and absolute log2 fold change more than 1. And second, we're going to need Cytoscape with different applications like a string EnrichmentMap Autoannotate and the gamma files layout algorithm to see those results. If you don't have Cytoscape installed in your computer, you can find it and install it. So once you download and install Cytoscape, you can open it. And next, you're going to install the applications that you need for this analysis. In the menu, you go to the apps, the App Store, and you can show the App Store. This search box will appear, and we're going to find the string application. Type a string, search, and we're going to go to the App Store, and we're going to install the string app. I already have it installed. If you don't have it, you will have them. This button will say download. You will download and install it, and we'll repeat with EnrichmentMap. Just type enrichment, click on search, and you'll see EnrichmentMap and install it, and repeat the process with the rest of the apps, like Autoannotate, same, and the gamma file layout, this one, and install it. So once you have all those applications installed, you will see them here: EnrichmentMap, Autoannotate, the gamma files, and the string database that will also have a enrichment. So let's go to the network tab here, and you will see that a string is already installed. We'll bring the list of differential express genes. Select all your differential express genes that have been previously cut off. Copy those. And you're going to paste that list here. Then you're going to go to these 3 lines and select the species. In this case, it's Homo sapiens, but notice that you have other species to select from. We're going to select the full string network, which implies that we're using not only the physical interaction between those genes or proteins, but also the functional interactions. We're going to use a confidence score that is median, 0.4, and we don't want to add any additional interactants. Once we have those parameters set, we're going to click on search, and a string is going to find the interaction between those genes, and it's going to display as a network. Notice that out of the 2,530 differential express gene, a string identified 1,824. So this is a resulting network. It has 1,124 nodes for proteins or genes in this case. And we're going to see the information here in the node table at the bottom, the gene symbol, and all the parameters that a string brings from the database, protein, etc. We also have 13,000 plus interaction between those that we can see here in the edge table. The string application can be controlled here. We're going to increase the size of this panel. Notice that you can control other ways that you visualize this network. For example, you can remove the colors, and you can do other like flat design, etc. So here you can do enrichment, functional enrichment. You're going to click on that button, and we're going to use, remember the background set I explained before. We're going to use a genome. Since this is RNA-seq analysis, we're going to use the whole genome as a background or reference set. We're going to click OK, and the functional enrichment is going to be run. The results will appear down here in a tab that is called string enrichment. You can increase the size of that tab, and you see that we have gene ontology, cellular component, biological process. We also have pathways, like reactome pathway, KEC pathways, etc. You can filter if you want to see only one type of process or biological process or pathway. You can filter by specific type. See, notice now we filter by KEC pathways, and you can see everything or not. You decide that. So notice that you can also export the results as a table for further analysis. As you can see, we have the type of signature, in this case, biological process, pathway, etc. Some identifier coming from the database, and gene ontology has this GO, etc. So that's an identifier. You have the name of the actual name of the pathway or the function. How significant is that overlap? The number of genes in the user list, in our list, that overlap with the number of genes in that pathway, and the actual gene symbols here. So we're going to go ahead now and cluster those pathways based on the similarities that they share in the number of genes that they have in common. So we click on the EnrichmentMap applications, and you can control the size of the clusters by moving this scale. This is a trial and error. If you want bigger clusters, you can move the scale to the smaller number. If you want smaller clusters, you can move the scale to the higher number. Let's put in the middle 0.5, and then we do the clustering. And as you can see, Now we have those pathways. Each of these circles is a pathway, and they are clustered based on the genes that they share in common. So if I click on this line that is connecting, I can go to the H table and see what are those genes that they are sharing in common. EnrichmentMap runs at the same time as AutoAnnotate. It runs in the background, and those clusters have been grouped by AutoAnnotate. You can see those clusters by clicking on hide shapes or not. You can see those circles. And you can also see the labels. They have been labeled. And also you can separate those circles to have a layout that is not so busy. You can separate in smaller clusters. You can control the way this visualization happens. You can say, okay, I want this specific color palette. You can decide not to use any, and you can color by a specific color or not. You can control the size of the border here, the color of the border, the size of the fonts, etc. So these names of these clusters come from here. If you go to AutoAnnotate application tab here, and these clusters have been grouped by the size, so have been ordered by the size of the clusters. In this case, this gated channel GABA is, remember that these samples coming from brain is expected to have pathways that are coming from the nervous system. And the way the name of these clusters is assigned is by finding the words that repeat more in all these pathways. I'm finding the word that repeats more and is added to the title. You can change those settings, so adding more words or less. But also you can change those. You can look and explore those clusters. The way you do that, you can zoom in, zoom out, or move around the network. Once you find a cluster, you can use these layout tools down here. You can select first the cluster in the layout tools. You can increase the scale to separate those pathways, and you might want to call it something different. In this case of GABA, yes, it's true, it's one of the most repeated, but it's about neurotransmission. I can say this is gated channel GABA, neurotransmission. So you can change those names to something that makes more sense biologically. Another thing that you can do, you can select these clusters, these pathways, cluster pathways. You can select this cluster of pathways and you can export those out of the network by clicking in this tool, select the nodal edges, and see here independently. So you want to maybe create a different visualization.